Symptoms. The most common visual signs of CTV are seedling yellows, stem pitting, and quick decline. The fruit on the tree begins to wither and/or produce low yields of fruit. The leaves will show clearing of the veins, cupping, chlorotic leaf flecking, and corky veins. Citrus tristeza virus, CTV, is a viral species of the genus Clostrirovirus that causes the most economically damaging disease to its namesake plant genus, citrus. The disease has led to the death of millions of citrus trees all over the world and has rendered millions of others useless for production. Farmers in Brazil and other South American countries gave it the name tristeza, meaning sadness in Portuguese and Spanish, referring to the devastation produced by the disease in the 1930s. The virus is transmitted most efficiently by the brown citrus aphid. Citrus tristeza virus, CTV, produces the most damaging disease of citrus worldwide and is a continuous threat to the citrus industry. It is primarily spread through infected rootstock and brown citrus aphid in a semi-persistent manner. Infected trees produce small-sized and poor-quality fruit, and in severe cases, tree dies. Sour orange rootstock is highly susceptible to CTV. Several mild and severe strains have been identified, which produce from invisible to visible symptoms in diseased plants. Existence of multiple CTV strains in nature is a challenge for CTV management. Symptomless infection in certain citrus species results in the inoculum buildup and further spread of the disease. Breeding for CTV resistance genes is a long and time-consuming process that takes 8 to 10 years before a CTV-resistant variety is developed. Genomic manipulation of CTV genome is challenging due to its larger genome size. Modern biotechnological tools can be used to control this disease and to prevent its spread in the future. However, CTV offers a valuable research tool for its role as a stable marker in genetic transformation of plants. This review highlights the challenges in developing resistant citrus cultivars against CTV and future prospects of CTV, and is an update to the research status of CTV, researched in 2023 in Pakistan. Trees infected with tristeza show light green foliage, poor growth flushes, and some leaf drop. The trees may produce a heavy crop of smaller fruit because the girdling at the bud union prevents starch transport to the roots. Feeder roots die from the tip towards the main root. CTV belongs to the genus Clostrirovirus, family Clostriroviridae. The family Clostriroviridae contains more than 30 plant viruses with flexuous, filamentous virions, with either mono or bipartite, one tripartite, single-stranded positive-sense RNA genomes. A recent revision of the taxonomy of the Clostriroviridae, based on vector transmission and phylogenetic relationships using 3 proteins highly conserved among members of this family, a helicase, an RNA-dependent RNA polymerase, and a homologue of the HSP70 proteins, led to the demarcation of 3 genera: the genus Clostrirovirus, including CTV and other aphid-borne viruses with monopartite genomes, the genus Ampulovirus, comprising viruses with monopartite genomes transmitted by mealybugs, and the genus Crinovirus that includes whitefly-borne viruses with bipartite or tripartite genomes. Viruses of this family are all phloem-limited viruses. Among the hallmarks of the group is the presence of a conserved 5-gene module that includes the 4 proteins involved in assembly of virions, the major, CP, and minor, CPM, coat proteins, P61 and P64, a homologue of the HSP70 chaperone. In addition, infected phloem-associated cells have clusters of vesicles considered specific to the cytopathology of Clostriroviruses. Although the evolution of the 3 genera of the family Clostriroviridae is unknown, it is interesting to note that dRNAs of CTV, analogous to each of the 2 RNA segments of the crinovirus genome, were found in infected plants, raising the possibility that crinoviruses arose. Understanding cross-protection by CTV. Examination of the ability of different isolates of CTV to prevent superinfection. By another isolate of the virus, CTV has long flexuous virions, 2000 MEAS-X 10 to 12 nm, that are encapsulated by two coat proteins. A single-stranded RNA genome of CTV, which is 19.3 qB, encodes 12 open reading frames, ORFs, Papu et al., 1994, Karasev et al., 1995. Figure 1. ORFs 1A and 1B are expressed from the genomic RNA and encode polyproteins required for virus replication. ORF 1A encodes a 349 kDA polyprotein that has two papain-like protease domains, plus methyltransferase-like and helicase-like domains. Translation of the polyprotein is thought to occasionally continue through the polymerase-like domain, ORF 1B, by a +1 frame shift. 10 3-end ORFs are expressed by 3 coterminal subgenomic RNAs, sgRNAs, Hilf et al. 1995, Karasev et al. 1997. Those ORFs encode the following proteins: major, CP, and minor, CPM, coat proteins, P65, heat shock protein,